I guess it's just you and me, Rosita.
Good morning. How are you doing?
Ah, okay. I'm waiting for the maintenance people to show up. I have a leak in the apartment.
Whoa, no.
Oh my God, Rosita, did you paint that behind you?
Yeah, yeah.
It looks so great. Is that your family or something like that?
Yeah, my family. I can bring it front and show you. I just
Is you in the picture? Are you in the picture now?
yeah, I am the picture, just this right beside my mom. I will
So
bring
you're
it.
the little, wait, the grown up or the child?
No, the grown up. Like, give me
Oh,
one sec. Give me one sec.
it looks really pretty.
Just wait a second. Before people, oh, before people join us, let me see.
Yes, it looks really pretty.
This is me. And it's not done. It's not finished yet.
No, but it looks really pretty. You know what it reminded me of far away? I know people are doing this. Um, you can, on certain websites, you can upload your picture and through some computing, they turn it into paintings. You know what I mean?
Painting. Yeah, I
You know
know.
what I mean? It looks really real from far. Yeah, yeah. You can tell it's a picture it's a painting from far away. It looks like a picture.
Yeah, you know, I use the just a knife to make it
Oh,
and
nice.
paint it.
Oh,
Yeah,
nice.
just knife and no brush because I want to keep it a little bit. What is this?
So you're doing oils, I guess.
A bit like abstract. It's not
Yeah,
abstract, but it's
I know what you mean.
just to keep it a little bit
And you're
uh,
using oils, right?
free. Sorry.
Oils?
Yeah, oil. Yeah,
Yeah,
yeah.
yeah, yeah,
I
yeah.
use
You can
oil.
tell.
Yeah.
It's really
I,
good.
I used to paint with acrylic, but I think oil is much better for me. So
Yeah, I used to paint with oils when I was a teenager. I don't do it anymore.
paint with what?
With oils,
With oil. Yeah, oil is awesome.
but it just takes so long to dry.
Yeah, but
That's the only thing. I don't have the space. I'm
yeah.
with the cat. He wants to
Yeah,
destroy.
actually, you can use your cat to paint. Just
Okay,
put his tail in the paint and then, yeah.
good, good.
Okay, so can I make you a main host? Do you want to be main host? Then,
Maybe I
so,
won't. Yeah, I don't mind. My internet connection has been good today.
oh, okay.
I mean, I haven't had any issues with internet connection or power at home. So I'm crossing fingers, right? Like I
Cross.
haven't had any issues.
Yeah.
But I'm home. I'm not at the office.
Oh, okay, okay. So we can keep you co-host as, I mean, whatever you
So
prefer.
if Caleb when Caleb shows up, we can make him the host. Is he
Yeah,
gonna
we
show up
can.
today? And we
Uh,
can remain as co-hosts.
sure, sure. Okay,
Just
so,
because I'm in the Wi-Fi, I'm not connected to wired.
mhm. Yeah, 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 yeah. So it may, yeah. Okay. So, yeah, oh, good morning. Good
Morning,
Good
morning.
Tom.
morning. Hi. Hi, Ivan.
How are
Hi,
you?
Rosita. Good
How
to
are
see
you
you.
doing?
Yeah, doing good. Doing good. Yesterday went really well, I think.
Yeah,
Pretty happy
actually, I
about it.
have heard the nice comments, very nice comments from your session.
Oh,
Yeah,
good.
that,
Good.
that
And yeah,
was,
and yeah, I thought Jim did especially well on Monday. That was
yeah.
great. Yeah.
So yeah, Caleb really liked your session, and he wants to go one step further for using MCell. So yeah,
Good.
he may contact you maybe later after the.
Yeah, good. Good.
When are you gonna
Morning,
have
Bob.
that? Oh,
Oh,
Hi, Bob.
hi.
Hi,
So,
Rosita.
morning, Bob.
hi. So what were you saying?
When are you gonna have the discussion about the Python version of MCell? Is it tomorrow or Friday?
Yeah, tomorrow.
Tomorrow.
Yeah. And Adam's gonna lead that discussion.
Okay, good.
Yeah.
Okay.
And Adam and I are still struggling with some small differences in the SBML files that are being generated by Cellorganizer.
Yeah, we haven't had a chance. Sorry, we haven't had a chance to respond to you, but I guess you could always manually edit them or something to.
Yeah. So I did that. But Adam and I are going to try to make our way through it today and figure it out today.
Great.
Yeah. So I'm pretty hopeful that we'll. Yeah. So there's Adam now. Maybe I'll ask him about that.
I'm going to go make myself a cup of tea. I'll be back.
Okay. Morning, Adam.
Morning.
I was just telling Bob and Ted and I guess Ivan about the SBML files that are being generated by Cellorganizer have those annotations in them, and of course we can edit them out by hand and just kind of have them available for people to load and try. We have that.
What is the issue?
So in the I'm not sure if there's an option in how Cellorganizer generates. Maybe Ted could answer if there's an option in how Cellorganizer generates the SBML files, but there are some CellBlender specific annotations that are placed, that are real, that are useful ones if we parse them and use them, and they're placed inside the parametric object block, where the point indices are located. The point indices are the indices that connect together the vertices to make the triangle faces. But those annotations inside the parametric objects block, for some reason, interfere with the element tree, the XML element tree module, Python module that's trying to parse the XML. And it's parsing the XML according to the description, according to the spec in the SBML spatial, level 3 version 1 official spec. For some reason, the element tree parser is not able to accept the annotations. It doesn't give an error. It doesn't give an error message when it's parsing. What you get is an empty parametric objects block with the point indices in it, so you get no triangle. So it's really
an example.
Sure. Well, Ted has Ted has the example, but I could forward it specifically to you,
Ted.
Ivan. Take a look.
Yeah, I can send it to you, but are you using like the Python XML parser, like the parse tree?
We're using the in the importer in Cellblender uses the XML element tree module to parse the SBML spatial XML file.
Yeah, he's trying to build a document object model from the actual XML file. And
right.
That parsing, it's something it's failing. It's so it could pass validation as an XML, but does not necessarily mean they will be parsed. So
right. And the parser doesn't give an error. It just returns an empty I guess component or something for the point index.
But it must have have a root, right? It would
there's a root for the whole, document, and then there's a root for the parametric object. Well, there's a parametric geometry block which contains all the parametric objects, and then each parametric object has in it the point indices, which is an unlabeled part of the block. It is the main part of the parametric object block, and then the parametric object block has that plus the vertices part of it.
Well, I don't think this is the only issue. I
Oh,
I think this
okay.
feature needs like a lot of more work. I mean, it's okay to show it as a demonstration, but
Yeah.
this will take much more time.
Yeah. I think probably it will. But 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 if we edit out those, it turned out that the model that Ted shared with us, that Bob and Ted shared with us, had what am I trying to say? I think it had 4 parametric objects in it, and or 2. And 2 of them had these annotations, and one of them did not have the annotations. And the one that did not, what clued me in what was going wrong was the one that did not have the annotations actually worked and generated the shape. And then I looked at what was going on, and I said, oh, the one that doesn't have those annotations on it. And so I went and edited out and removed the annotations from the other 2. And the annotations are only like 3 lines of text, opening and open annotations tag, the content, and then the end annotations tag, right? Those 3 lines, I just edited those out of the 2 parametric objects, and then those worked as well, and it was beautiful. It looked great. So I'm sure that that's what the issue is with those.
Okay.
Yeah.
Yeah, I can look into that as well.
And what I then once I found that, then what I did was I looked online for how to tell element tree to ignore tags. If there's a way to say just here's a list of tags to ignore, don't worry about it. And I couldn't find a good set of documentation for how to do that. And then at that point I got kind of lost. Anyway. But we're supposed to be having our icebreaker now, so let's have our icebreaker, and then Adam and I can, and all of us can fight with this a little bit later offline.
Okay, so it's going to be icebreaker time, yeah?
Yeah.
Yeah, who wants to start? I mean, the coffee hasn't started working on my brain yet. So feel free to start.
Yeah.
Yeah. Okay.
Well, there's a few people on here who we hadn't heard from yesterday, introducing themselves. So,
so what kind of icebreaker did you do before? Just a general.
Yeah. So yesterday we had, on Monday, Terry put us all into breakout rooms and had us in small groups come up with the ultimate question of life, the universe, and everything that could be simulated in biology, right? That could be simulated, and or we would like to ask God the question, what's going up with this thing or that thing, right? And that was a lot of fun. And then yesterday we just had people go around and introduce themselves, a few at a time. And so we could continue that today a little bit. If someone wants to volunteer and tell us about themselves, maybe Ivan, you can, since you haven't been on here before, maybe you can start and tell us a little bit about yourself.
I mean, I don't know if Bob and Ted want to start since today is their day.
Sure.
Oh, go ahead, Ivan.
Okay, so my name is Ivan, and I'm a research software specialist at Pittsburgh Supercomputing Center. I'm part of the PSC team. But before that, I used to work for Dr. Murphy, for Bob, for 10 years or so, a long time. And I was one of the main developers for Cellorganizer before I joined PSC about a year ago. I'm still involved in MBIOS, but in a different role. But I'll be here today, not only providing support to PSC, but also to anyone who has questions about Cellorganizer. So that's about my introduction.
Okay,
So
perfect.
hear from any of the people from the participants we haven't heard from before?
I'm not sure if we heard from Randy yesterday. I'll single out Randy if he's really on there.
Sorry. Yeah, I thought a lot in the chat. Hi, Tom. That's good to see
Yeah,
you.
yeah, you did. You said a lot in the chat, that's for sure.
Yeah. Yeah. So I don't know what are we supposed to like talk about?
Well, just say a little bit about yourself.
Oh, okay.
Yeah.
I'm an assistant professor, cell biologist. I do cellular neuroscience, mostly with imaging approaches. And I study gap junctions, mostly gap junctions of astrocytes and the gap junctions that form between astrocytes and oligodendrocytes. I have dual screens. That's why I'm not looking at the camera.
Yeah.
And I'm in my garage right now. So that's where all the good things happen. So
Yeah. I couldn't
yeah,
tell
so
if that was a
I'm at the
garage
New
or
York
a lab.
Yeah, I'm down in the garage today. I'm not in the lab, but
Yeah.
I'm at the New York Institute of Technology, which is on Long Island. It's about a little under 10 miles from Cold Spring Harbor Laboratory, and further west in Stony Brook, some of the other institutions there. What else? So I've been trying to use and using, I should say, a cell blender for several years now. I attended one of the workshops at Pittsburgh, and that's how I got into this and realized I needed to learn how to use Blender and went from there. And it's been really exciting, and it's been really exciting to see how much effort and success the development team has had. This workshop is really great. I'm learning so much so far, and I'm looking forward to the next few days. I'm wanting to use Cell Blender to simulate different aspects of gap junction and astrocyte biology. So we've been working on a model for a long time to look at whether or not intercellular communication is altered when the channels are moving in the membrane, and whether or not their proximity to organelles, other organelles such as the ER and mitochondria, is altering the communication that's achieved through the gap junctions. So that's the goal as far as MCell and Cell Blender go. And because I do a lot of imaging from the molecular single molecule level up through to whole tissue imaging, I'm interested in combining those data into models all the way down to BioNetGen up through at the single molecule imaging level and doing single molecule tracking all the way up to looking at the variation between cells using more wide field imaging and confocal 3D imaging. So that's my area and my reason for attending this excellent workshop. I'm always so excited to see everything that everybody's doing. So.
Cool, thank you, Randy. And about your gap junctions, I think maybe Adam can fill me in if I'm wrong, but I believe we have a way for receptors on one membrane to interact with receptors on a separate membrane.
Oh, that's cool. So the way we've been implementing it was originally back when I attended the previous workshop, you created this kind of like offset teleportation across the two membranes.
Right, I remember that very well, Randy. Yeah.
And then in between then and now, a PhD student and I made a model. Now that it doesn't have to be manifold, we made a model with just kind of like a cup on one side, and then it was coming through like that.
Right.
Or we made a transparent membrane right there, region, a surface class or something. I can't remember which was our solution, but
sure.
That sounds awesome, because if you can get that docking procedure and then have them move in relation to each other, that would be really interesting to try that version out.
Yeah. So Adam, so after they dock together across the membrane, can they diffuse together in tandem?
Not currently, but Joe and I, our cooperator in Israel, they needed them to diffuse. So
Okay. So yeah, so we're working on that. So
right
yeah.
now they like must stay at the same place, but yeah, we will implement diffusion for them as well.
Yeah.
So one of the things that brought me to MCell in the first place was I saw your group's work with Gina Sosinski at UCSD, looking at the myelin sheet. So
Yeah.
in between then and now, there's been a lot of publications, and there's this idea that there's this xenotypic, so-called xenotypic coupling between a connexin on one membrane of the myelin sheath and a KV channel on the axonal membrane at the parajuxtanode. So
Yeah.
it'd be really cool to kind of pick up where you guys left off on that really, really detailed model to build onto it with the new biological knowledge that we have.
Yeah. And actually, what that case you just mentioned is one of the things we had in mind when we started to designing how to make this work. So absolutely, yeah.
I'm looking forward to hearing more about that, Adam and Tom, about the things being able to move in tandem, diffused in tandem like that. Yeah.
Okay, yeah, so we'll let
Thank
you
you.
know about some news in that area.
Cool.
And I'll just in general say about this workshop is each of our teams has just the one day to present and teach you about it, and then the advanced topics. But really, there's really not enough time. We're just scratching just scratching the surface, right, on all of these things, and there's much deeper that one can go with this, but not even in a 5-day workshop, right? Can you cover?
And yeah, and that's a good point, that we always stand ready to help people after the workshop. If you have, questions or even new features that you'd like to see, just let us know. I mean, that's part of our job, is to help support the community.
Yeah, that's our mission statement. Okay, I don't think we heard from Susan Mertens yesterday. Maybe if Susan's on, maybe she will feel comfortable to say a few words about herself.
Thank you. I just came on.
Oh, okay.
Okay. I am a faculty assistant professor at Mount St. Mary's University in Emmitsburg, Maryland, and also a guest researcher at the Frederick National Lab for Cancer Research. And my interest is in rule-based modeling in DNA damage response. And so we're just starting those models in BioNetGen, and so this has been really great to get updated. And so my actual interest in using those models is with the ATOM research, well, it was the ATOM Consortium, now the ATOM Research Alliance, that is trying to do drug discovery with machine learning, and hopefully with models from RuleBender.
Oh, cool.
So I wasn't here as well, I think, yesterday at the Icebreaker.
Okay.
Let me just, yeah. Hi, so I'm Wouter. I'm a PhD student in the lab of Harold McGillivray in Utrecht in the Netherlands. And we study the nanoscale organization of the synapse using super resolution microscopy and electrophysiology. And yeah, it's been really great so far, I think. Learned a lot of stuff already, and I'm mainly interested in using Cellblender and MCell to simulate all the findings that we have basically in the nanoscale distribution of proteins in the synapse.
Okay, awesome. One of my passions is exactly what you described, Bartek.
Good to know.
How about Sejin? I'm not sure if I'm pronouncing that correctly. Sejin Gulek, if you're there. Uh-oh. Oh, there he is. Hi.
How are you? I introduced myself yesterday too. I'm currently going to be a PhD student in the University of North Carolina. And I'm really enjoying the workshop so far. And I really liked your presentation yesterday. I looked about, I knew about Blender. I saw a lot of videos on YouTube, like people doing special effects and those kinds of stuff, but I never seen being used for like biological simulations. And it was really exciting. And,
oh, great.
Mhm.
Yeah, there's a number of groups across the internet that have been using Blender for simulating, biological things specifically, and also in general scientific visualization. It's a really powerful environment.
Yeah.
Let's see, how about Del Valeria?
Oh, hi. Can you guys hear me?
Hi, Del.
It's close to midnight here in my time zone.
Oh boy.
Yeah. Okay, so I'm an undergrad student studying biomedical informatics, dual degree from Edinburgh and Zhejiang University. And my friends and I have been using MCell and BNGL since last summer to model viral absorption. And I found the workshop really helpful. Naturally, we've been able to apply some lessons that we learned yesterday and 2 days ago to our project. And hopefully we'll learn more in the advanced topics tomorrow and the day after tomorrow.
Okay. I'm not sure if we heard from Maurice yet. Maybe we did, but not remember.
No, I think not. Turn on my camera. Right, can you see me? Yeah. Hi.
Yes, hi.
So I'm a PhD student from at the University of Göttingen in Germany, and I'm mainly interested in receptor trafficking at synapses, AMPA receptor trafficking, and also in the dynamics of the PSD. And yeah, so I came across MCell when I was looking for tools to simulate AMPA trafficking and also like dynamics of PSD molecules and so on. So I'm was also looking forward for this workshop to find out more about how to use MCell and what you can do with it. And actually, I'm also interested in like this liquid-liquid phase separation that is has got a lot of attention recently. So I'm looking forward to these new implementations that you said that are planned, where you can then also have maybe interactions and volume exclusion between molecules. to do this kind of stuff. Yes, yeah, that's from my side.
Okay, thank you, Marie.
How are things in Göttingen? How is the pandemic winding down?
Oh, it's quite good at the moment. In Göttingen, I don't know what is the situation in Göttingen compared to the rest of Germany, but at the moment, it's quite good. So vaccination is going on, so I think we are at like 58%. So that's good.
Yeah.
For 10 years prior to the pandemic, I'm an honorary professor at the University of Freiburg, and so I've been going 2 or 3 times a year up until the pandemic, and of course not since. So I miss Germany and Freiburg quite a bit.
Okay, it seems that we have only 2 minutes until the workshop, the main session starts. So does anyone else want to introduce herself or himself? Okay. Rufaida, I know your name from the other workshop, from hands-on computational biophysics workshop. Am I right?
Yeah, yes, Rufaida. Yeah, hello, Rosita. Hi, everyone.
So
I'm fine. I'm fine. I'm doing great. Thank you so much for these wonderful workshops. Probably these workshops are not very much related to my research area, but I learned a lot. I learned from the Blender yesterday, and that was really amazing. And I hope that I can learn something new. Yeah. Thank you so much.
Sure.
Do you want me to introduce myself? Okay, I'm sorry. Yeah, I'm an assistant professor at Jordan University of Science and Technology. So I'm from the Middle East, and I'm a medicinal chemist. My work is mainly computational modeling on G protein coupled receptors. I like to do MD simulations to understand the molecular basis of signaling of these GPCRs, and that's all.
Thank you very much.
Thank you.
So on that topic, Rufaida, what are the prospects for getting good kinetic rate constants for G protein coupled receptor signaling cascades?
Sorry, I didn't hear very well.
My question was on the topic of G protein coupled receptor systems. What are the prospects for being able to get good values for the kinetic rate constant for those interactions?
Okay, it's 11. I mean, Eastern time, sorry, 8 Pacific time. So, okay, Bob, do you want to start?
All right. So before I get started, I just want to emphasize that I love being interrupted when giving a talk, because that means at least someone is paying attention. And so please don't hesitate to just interrupt me I'll try to stop every so often, but if you have a question on a particular slide or a particular point I'm making, please just go ahead and you don't have to put it in chat or anything. Just unmute yourself and ask away. All right. So I used to be the head of the computational biology department and the School of Computer Science at Carnegie Mellon. That I started that department something like 13 years ago, and I was the head until last year this time, and I just retired as of this month, but I am still quite active in my research program, and my special interest is in modeling of the way cells are organized, the spatial relationships between different components of cells. So what we're going to talk initially about is the basis for our project that's been going on for many years called Cellorganizer, which is, with the goal of which is to build models of how the various components of cells fit together. So this, of course, is a major problem, right? Cells don't actually look like the cartoon on the right, and every cartoon you've ever seen of a cell is wrong in so many ways, including the fact that things are not put in the proper spatial relationships. They're not put at the right sizes, so that the diagram here of the different components are all shown roughly similarly, but they're nowhere near as similar in size. And fundamentally, that most of them don't actually look the way those things are drawn. And so this is a problem that that the world has been studying, the fields has been studying for hundreds of years, and we are still not to the point where we can capture a complete model of, for even one cell or one cell type, how all the different organelles are organized and all the different structures are organized within the cell. And even somewhat straightforward things like the variation in size and shape of different cell types. So these are all questions that I believe need to be answered with a combination of microscopy and machine learning AI to build models of how cells are put together that we can't learn from just individual pictures of individual cells or even pictures of individual tissues. And so the fundamental questions that we're trying to address are, how does all of this get put together relative to each other? How the pieces put together relative to each other? And how does that vary within cells, between cell types, and over time, such as during development? So there's a major field, which is a major component of cell biology. And I think the difference in our approach is to trying to approach this as a computational machine learning problem of how do we represent all of this. And just to make the point about how things don't actually look the way we think they do, here are some examples of cartoons and the corresponding example fluorescence microscope image. This is for the Golgi. This is also a case where notice the sizes of these don't correspond to the sizes in the cartoon, and these are lysosomes. There are lots and lots of lysosomes spread throughout the cell, but they do have a sort of a semi-predictable pattern of being mostly near the nucleus. Here is the microtubule network. Very many of you are familiar with this. Here is the mitochondrial network, which is shown, typically as isolated mitochondria, but it is a combination of isolated mitochondria and networks of interconnected mitochondria. And so the way that cell biology has been working for at least a century is that people do some kind of experiment, collect an image such as the ones we've just been looking at, and then write down what they think that is showing. And so they do some imaging experiment and then convert that primary data, they interpret that primary data into statements like, these secretory vesicles that I labeled bind to microtubules, or, these lysosomes are mostly located around the nucleus, or, you do a biochemical experiment and you say, when I, isolate, vesicles of a particular size, they have, particular properties, whatever it might be. And all of that gets put down in words traditionally. And the problem is that it is quite difficult. A problem is it is quite difficult for complicated organelles and to come up with words that adequately describe the relationships, or that even describe them reasonably. And they, of course, don't adequately describe them. And what do I mean by adequately describe them? Enable me to imagine, even just in my mind, synthesizing what a distribution of a particular component might look like. And so those are challenges that we're going to try to address, and that we've been addressing with Cellorganizer for, almost 20 years now. And so those are our goals, and our goal is to try to do better at that task using machine learning. And a starting point for this was work we did many years ago, which was to describe the cell patterns with numerical features, and then use those features to say, this is the pattern of lysosomes, or this is the pattern of endosomes, or this is the pattern of mitochondria, in a classification approach, a pattern recognition approach, using those features. And that is now widely used in packages, such as those here, for describing what is going on in the distribution of a particular fluorescent probe, within a particular cell population, looking at whether two cell populations differ in something, look at whether or not, adding a drug changes the pattern, and so on. But the fundamental approach there is recognizing a pattern, classifying a pattern, or describing a pattern with numerical features. But there are two major issues there. One of them is, and anybody that has done this kind of work realizes, is the features that one calculates are tied almost always to the particular microscope, objective, cell line, etc., that was used for collecting data. So when using any of these kinds of approaches, it is often the case that everything is kept the same, same microscope, same cell line, and now we're just going to look at one variable that we're going to change. But that doesn't help us in terms of trying to figure out how to put all of this together. And it also doesn't help us in terms of being able to use a classifier that, someone at one lab builds, to use that in another lab, because the images of the second lab may not be collected in the same way. So, and there are, of course, lots and lots of ways in which we can collect images. And when we use these descriptive features, those are inherently tied to the properties of the microscopy that we're doing. So what that means, if we, for example, do something like so-called high content screening or analysis, where we have some negative control images like cells without a drug, and then positive control images with cells with a particular drug, and we calculate features and learn a model that distinguishes between these two, we can't apply that model to images from a different lab, different screen, different microscope, and so on, okay? And that's because of this nature that the features are tied very heavily into the way in which the microscopy is being done. Now, the other thing that we need to consider here is that these types of descriptive approaches are only needing as much information, as much in the features as needed to tell the difference between two different things. So if you are trying to look at pictures of apples and oranges and decide whether a new picture is an apple or an orange, you don't need to know a whole lot about apples and oranges. All you need to do maybe is to know the color, or to measure the roundness, or measure whether it has a stem or not, or whether it has an indentation in where the stem is. So you can answer the question for a new image of which of these classes it is with very simple and a small number of features. However, if someone says, okay, here are a bunch of images of apples, these are apples, here are a bunch of images of oranges, here's an orange, these are oranges. You can train something to recognize apples and oranges with a very simple approach. But if someone then takes those away from you and says to you, what does an apple look like? Draw an apple. That can't be done with just those one or two features, unless you want to say that an apple is just a red blob, okay? So you need more information in order to be able to reproduce or to generate a new example of a particular pattern or object. That is a generative model, okay? So we distinguish between discriminative models, which have just enough information to be able to tell the difference between A and B and C and D, as many things as we want, versus a generative model that tries to fully capture the characteristics of the things that we're working with. So you are all familiar with generative models. Anybody that's actually anybody that's been doing any deep learning is also very familiar with generative models. When we were children and we were learning our numbers and letters, we were given examples of numbers and letters, and we could have tried to memorize every 5 that we saw. And if somebody asked us to draw a 5, we would maybe just reproduce one of the ones we memorized. But that's not what we did. We learned a mental model that is of what a 5 really is, right? That allows us to generate new examples that are not the same as any of the examples we have been shown, right? That's what we do. And that's the basic idea of a generative model, that we are learning the characteristics of the thing such that we can reproduce it, and it will be, statistically, we would say the generated examples would be drawn from the same distribution as the examples we trained from. Now, that's what we want to do with images and cells. Okay, so again, we'll take some training images, and for example, we would be told, okay, these are images of the pattern of lysosomes, and we're going to try to build a statistical generative model that allows us to generate new images that are similar, that are statistically similar to the ones that we learned from, using as much information as we can from the images that we're training with. I'm going to pause for a second. Are there any questions about anything I've talked about so far? Very philosophical introduction. Questions? Okay. So one of the major advantages of generative models is the things we extract, what we would call the parameters of the generative model, are not directly tied, in most cases, to the kind of imaging that was done. Because what we're trying to capture there is, how do I regenerate this thing? And as with the example of fives, I might be given different examples of fives that are different sizes, different colors, different pixel resolutions, high resolution ones, thin ones, thick ones. When I'm learning that model, I'm learning what is the characteristics of a five, such that I can learn that from examples that were from different types of images, different input. And the same is true when I'm learning models of images. I can have images that are from different microscopes, that are different resolution, that are even different types of microscopy. And if what I'm trying to do is construct a model that is going to capture all of that and be able to generate new examples, then the parameters of that model that I learn are generalizable. They are not tied to a specific type of microscopy. And that's the goal. And in that case, if someone at one place learns a generative model, that generative model should be applicable as well to other microscopy. Okay. And it means that if you build a model over here, you can directly compare the model. Let's say you have some data over here, you build a model. You have data over here, you build a model. You can directly compare the models without having to compare the images themselves. Does that also make a clear concept? So major advantage of building generative models. Okay, so that's what this Cellorganizer project is about. And the idea is to take in cell images and then build statistical models that capture different aspects of the incoming model so that we learn the characteristics of those things and we can combine them to produce a new image, such as the example shown on the right. All right. And this part is going to change depending on what it is what aspects we're trying to build and how many and where the project has evolved to. So we have lots of different components that can build different parts of these models and that can be combined. But this is just an example of a particular model that one could build that would look at nuclear and cell shape. The arrows here are showing dependencies, we would call them, that the cell shape depends on the nuclear shape because or you could look at it the other way around, because the nucleus has to be inside of the cell. So you have to know where the cell is to know where the nucleus is, or vice versa. And the same thing is true for any objects like organelles, mitochondria, endosomes, and so on. We have to know where the cell and nuclear boundaries are to know where those objects are, or to know what the distribution of microtubules are, and so on. Okay, so that's the concept we're going to start working with. And that's the homepage for Cellorganizer. Now, so another way of saying what I described to you is learning the assembly instructions of a cell. So we can think of this generative model as, if we had all of the components of a cell, how would we put a cell back together? If we had little tiny models of all the pieces of a cell, how could we, assemble a cell back together? And that is basically the same paradigm as building models. You know, all of you, I'm sure, have done some kind of modeling when you were younger, or most, with things like Legos or so on. There, you're given the parts, and what we're trying to do is to finish the model. The instructions are inherently hierarchical. They tell us to do step one, put something together, and then do step two, and then maybe step three is put the two pieces that we made before. So, you know, here's an example from Legos, right? It says put these pieces together like that, then put this together on top of that. So that's where this hierarchical concept comes. And that kind of model is a forward model. We're given the pieces and the assembly instructions, and we're going to build the cell. Okay. Now, we could also take a finished Lego model and take it apart, and if we do it carefully, we could regenerate the instructions, and we might not generate them exactly the same way, because we might take the assemblies apart to different extents in a given step, but we would be able to take them apart, and then, given the instructions, if we carefully write down all the steps we followed, we can put it all back together using our instructions, even if they're slightly different in the order or the division of things from the assembly instructions that came with the kit. We can learn the assembly instructions by taking the model apart. Now, we can do that for Legos because that is a deterministic process if we are following the instructions. That we always end up with the same thing. So from that perspective, you know, Lego models, you know, are a little bit different than putting, you know, a furniture together, you know, IKEA furniture or something, where sometimes you don't end up with the same thing every time. Now, in contrast to that deterministic kind of model, cells are probabilistic. Any two cells don't have all of the components, all the parts in exactly the same places and exactly the same shapes. But we can still think about the process of putting cells together as this hierarchical assembly process, okay? And incorporate the probabilistic nature into that model. And the machine learning concept that captures this very well is the concept of a graphical model that we're going to talk about briefly, okay? So what you can see is here's where we're starting to work towards this fundamental cell modeling question of how do we put a cell together, all right? So what we're going to do is something analogous to the way Lego instructions works. We're going to say that in our graphical model, we're going to have a node that the node contains the probability of finding a particular part somewhere within the cell, okay? So for the example of lysosomes, we might have a probability distribution that says it's mostly near the nucleus, but there's some probability that it's further away. So each node is going to contain that probability distribution for a particular part or assemblies of parts that we made previously. Then we're going to have edges that are going to correspond to those dependencies I mentioned a minute ago, right? They are the pieces that are needed, the things we need in order to know where and how to make a new assembly, all right? And so the edges indicate what nodes do I need in order to make a new node. So I want to put a new assembly together. I'm going to have a new probability distribution for that assembly. what are the things that I need to be connected to in order to know how to put this one together? And then we're also going to have what we would call a node function, which tells us, given the inputs defined by the edges, how do we make the node? How do we put the pieces together? And you can think of that in the Lego sense that the instructions don't just say, put the blue part, together with the gray part. They tell us how, which way to arrange the two parts relative to each other. All right, so here's a simple example of how we would construct a graphical model from Lego instructions, or from disassembling a Lego model to build the instructions. We have our two parts, and what we're going to do is we're going to make a node for the gray part, and we're going to make a node for the blue part, and we're going to say the distribution of the blue part depends on the distribution of the gray part. Okay. And what is it that lives in these nodes? Well, this is a probability density, a two-dimensional probability density of the gray part. This is saying, here are the places where this gray part is, okay? You could also think of this as being represented as just a single value here. If you knew the shape of the gray part, then you would know there's one centered right here, right? So this node, this is just for illustration, this node is holding where's the gray part in this space. And then we would apply our node function to generate the distribution of the blue part, all right? And the node function says, put the blue part on the little, corners, if you will, of the gray part, all right? And that produces the distribution of the blue part, which we would store in that node. So, of course, this is a deterministic example, but that's the basic idea of trying to construct a generative model by constructing a graphical model that tells us what depends on what and how do we put things together. All right. Now, I make this point because part of the idea of this workshop is to help maybe guide you guys in thinking about this overall problem, not just in using Cellorganizer or not just in using all the tools we're talking about in the workshop, but also thinking about how some of these concepts might be relevant in your future research. And,
excuse me.
Yes, please.
Would you mind showing the previous slide? I'm just curious, what was the definition for edge?
Right. So it is it connects nodes to nodes that they depend on. So that's what this arrow, that's the edge, and it's a directed edge, right? It's saying blue depends on gray. And
Okay,
if we had,
if
we had other one, we might have, for example, that blue depends also on some other piece that like it would only, if there was a piece here, you would only put the blue ones near that piece. Okay.
Mm-hmm.
All right.
I see. Okay, cool. Thank you.
Other questions about that? Thanks for asking. Okay. So initially, we constructed generative models of cell organization by just considering one organelle at a time, very simple models. And what we had was we would construct models of the nucleus, the shape of the nucleus, models of the shape of cells, and then models of where within the cell was a particular organelle located relative to the cell and nucleus. So that's a generative model, a graphical model, where we would have in this case, we would construct it so we would build the nucleus first, then have a model that tells how the shape of the cell depends on that, and then a model of where the organelle is relative to those two, okay? And we also constructed models of how to grow a microtubule distribution if you knew the cell and nuclear shape. Okay, so those are some very simple graphical models, illustrations. Okay. Now, when we do, so to connect to the work that you've heard about with yesterday, with doing cell simulation, well, the last 2 days you're doing cell simulations, you learned about compartmental models. And in many cases, when doing cell simulations, we use a cartoon compartmental model, like draw circles or spheres and say that is representing the compartment. And we can learn what the distribution of any protein that we are studying. So, you know, we can look at proteins as markers of organelles to learn about the organelle, but we can also learn just about the protein itself and where it goes within the cell. So, you know, when we do cell simulations and they involve reactions between proteins, we can, you know, use a cartoon model and put proteins into it by saying there are so many molecules or such and such a concentration of a compartment to put that protein in. And we can even use real images to determine, you know, the surface area or volume of particular compartments. And then we can do all kinds of different simulations that you've heard about, whether using differential equations or using particle methods. The issue here is the geometries, that if we draw the geometries, you know, as these kind of cartoons, the geometries themselves are not very realistic. And so that is also something we're going to touch on and when we're talking about using cell organizer models. And so to summarize the kind of the concepts that is driving the way we're doing this, unlike literature, you know, where you collect some images and then you look at them and then you decide to write some words and, you know, then those words become the output. Here we're talking about automated models. Everything is learned directly from images. As we said, they're generative so that they are capturing, ideally, all of the aspects that were used to put in to build the model. That they are statistically accurate, that they are not just an average cell or an average distribution, but that they capture the variation in a distribution from cell to cell, and that they are also compact, that they are not just memorizing examples, but rather that we are learning these parameters or the characteristics from the examples in order to have a more compact model. And this is where we're, you know, talking about this inverse problem, as I mentioned, not a forward problem where we have the parts and the instructions we build the model. Rather, we're given the complete cell, and we want to, and we know the parts, and we just want to learn the assembly instructions. This is different from just reconstructing a model from images where what we're looking for is, can we get rid of any imaging noise or variation, for example, from cell to cell or organelle to organelle or ribosome to ribosome? So there's a lot of work that has been done on this problem of reconstructing, either by, as I said, you know, estimating what the underlying structure is by removing noise, or taking lots and lots and lots of images of different ribosomes or other structures and averaging them so that you have an average model. So that's reconstruction. we're interested in reconstruction to a small extent in trying to estimate the parameters of our model, but we want to keep all that noise, all that variation, okay? We don't necessarily want to keep the noise, but we want to keep the variation that is the biologically meaningful or biochemically meaningful variation that occurs from organelle to organelle or from cell to cell. Okay.
Excuse me, sorry,
Yes,
I
please.
have another question.
Sure.
My question is about the cell shape. So I'm just curious, for example, if I have 100 cells with different shapes, I mean that just for example, from a rounded shape to a protrusive shape. And so by using this kind of pipeline, can I identify them can I classify these cells based on their shape, or I can have the features, for example, in which feature they are different?
Yep. It's a
So
great
which
question.
ones?
We are going to talk a lot about shape. And so I'm going to answer briefly now, but when we come to it, maybe if it's not clear, you can ask the question again. But let me just start by saying the way most people work on that problem is to calculate a very small number of very simple features like the length or the width or the area of a cell and ignore everything else. And in many cases that will work. So if you're trying to tell the difference between, lymphocytes and fibroblasts, the size will help you. The lymphocytes are typically rounder than fibroblasts and so on. So there are very simple features you could use if all you want to do is to distinguish between two cell populations. But in many cases, and we'll see examples of that, we don't just want to distinguish A from B. We want to understand what is the variation and why is the variation there within some cell population, or how does it change during cell differentiation and so on. All right. So we're going to try to capture generative models of cell shape instead of just discriminative models of cell shape.
And it means, yeah. And it means that Cellorganizer is based on the deep learning approach instead of defining the features. Is it correct?
It doesn't have to be deep learning. We certainly use deep learning in some cases, but it doesn't have to be deep learning.
Maybe I should say.
Yeah, because you can learn. You
Yes.
can learn how the shapes are varying without having decided ahead of time how they're varying. And we're going to talk a lot about that.
Okay, so maybe I should be patient.
It's fine. Yes, but please, when we get to it, if it's not clear, please ask then. Okay?
Yeah, sure. Thank you so much.
Yes.
And Bob, there was another question from Dima. He's asking, can you comment on Cellorganizer's ability to view patterns hidden or blurred due to poor resolution in a real fluorescent image?
Yep. So what we have is another good question. So when I was mentioning that it's independent, largely independent of resolution, it's also largely independent of noise. But of course, if I have a completely blurred image, I can't learn anything from it. So the point is that we are doing a kind of reconstruction in the initial estimation of the parameters of the cell, where we are trying to remove noise, or we are trying to capture a particular size or shape independently of the resolution of the image that it was acquired. But there are limits to that. So if we have images where cells are 2 pixels wide, then we're not going to be able to do anything. Okay. But if we have high pixel resolution with some blur due to point spread or whatever, we may be able to reconstruct individual components reasonably accurately. And because we're doing that over cell populations, we may do a better job of learning the overall properties, even if there is noise present. Did that answer your question, Deepak? Well, okay. So here's where we're going to talk about deep learning briefly. Traditionally, these kinds of problems, these are parametric models that we're talking about. They've been done by deciding what the parameters should be and then learning them. And the hard part then often is deciding on what the parameters should be, deciding on the techniques to be used for reconstructing the model. And that is the approach that is largely used in Cellorganizer, this idea that you have images, you parameterize them, you convert them into parameters, and the parameters are such that you can reconstruct the original image from the parameters. And then you build a statistical model that tells you how the parameters vary in a cell population. And that statistical model can be sampled from to give you new parameterizations, which you can use to synthesize new images. Okay. And that's the approach that we're going to use, with some modifications I'll talk about, where mostly we are going to choose methods to do this parameterization. Okay. But of course, you probably all know about deep learning. If you have large numbers of training examples, then we can learn directly from images. We can build a probabilistic model directly from images and synthesize images from that, okay? Using things like autoencoders, for example. And there's a great study that was done a couple of years ago by actually one of my former students, Greg Johnson, where you can actually learn generative models to predict what fluorescence would look like from just a bright field model, or just a bright field image, okay? But there are some problems. Learning these kinds of models from using deep learning take lots and lots of images, okay? And that can often be a problem. In many cases, it's not. So if you have lots of images where lots might be hundreds to thousands, or for many applications, millions are needed in order to learn good models. But so that can be a problem. But even if that is not a problem, there is another, which is that deep learning models, they only understand of images, they understand pixels. It is very hard to get them to understand structures or objects. Okay, and they are also, depending on how they are trained, they are often not easy to compare across images from different modalities. There are techniques that are used to try to improve that. And so when we generate cells with deep learning models, you know, what you end up with is a probabilistic distribution of a protein fluorescence or something like that. And cells are not made of blobs of fluorescence. They have typically discrete organelles. And deep learning models in general don't learn those boundaries in a way that they can synthesize. There are exceptions, and you know, I'm happy to talk more about this towards the end. And we also use deep learning models for a lot of what we do. But what I'm going to focus on today is on using parametric models because they can be trained on much smaller numbers of images and can be trained to do trained to work better in many cases. And you know, just to come back to this study, I don't know if any of you've seen this study before, right? The idea of this study was to take bright field images paired with fluorescence images, train a deep learning model that would learn to predict one fluorescence from a bright field. So you have pairs of bright field images and fluorescence images. You learn a model to predict the distribution of fluorescence given bright field. And then you do that for another pair, another set where you have pairs of bright field and a different fluorescence and so on. And then you apply the models you learned separately to the same bright field, and that allows you to produce an image that has all of those organelles in it. But there's a problem, and that is that what often ends up happening is the model predicts the same organelle in the same place, or multiple organelles in the same place. And this just illustrates some characterization of that. This is measuring how exclusive the distribution of each organelle is relative to other organelles. So how much overlap is there in the synthetic images of organelles using deep learning? And, for example, desmosomes, they overlap, they're generated such that they overlap with almost everything. And so the scale here is all of these are not overlapping very much compared to desmosomes. But if we blow the scale up, what you can see is that that some of these organelles, the ones down here, have very low exclusivities. That is, they overlap with each other when they're being in the synthetic images. So this is addressing the issue that when you want generated generated cells, you want them to have the characteristic of cells, which is that organelles in general don't overlap. All right, so we're going to talk about another challenge. So fluorescence microscopy is great for learning about cell organization and cell shape and so on. But we can typically only measure a small number of molecules, organelles, probes, whatever, at one time in live cells. That number tends, I think the largest that's ever been done, I think, is 6 or 7. And of course, there are a lot more than 6 or 7 proteins in a cell. And so the question becomes, how do we combine information from different images? So if we can only do 5 or 6 or 7 at a time, how do we combine information when we've constructed models from different subsets of molecules in different images? How do we combine them? And again, what we're going to try to do is to combine them through generative models, where the generative models, like the example we just showed with bright field, generative models built on a common reference, okay? And there are 3 examples we're going to talk about with respect to this, where what we're going to do is we're going to take separate images of different organelles and try to learn their relationships to each other using generative models on a common reference. And the first of those we're going to talk about is punctate organelles. So this is an image from the Human Protein Atlas collection, where the green shows the fluorescence of a particular protein, Sec23b. Red shows tubules, microtubules, and blue shows the endoplasmic reticulum. And what happens in the Human Protein Atlas project is that they label many different proteins. And then they have a panel of human annotators that look at the pattern of each of the different proteins, and they assign words to describe them. And they actually ran a competition a few years ago for trying to do that using deep learning. And it surprisingly did not do that well. It does reasonably, but not, it can't, for example, do this problem that I'm showing you here. And I'll tell you why in a minute. These are 11 different, each of these cells is from a different collection of images. These are for 11 different proteins. So these are all proteins where they were labeled and people were shown the image, and the human annotators said, I can't tell the difference between these things. I'm going to call all of these things vesicles. And the question is, can we do better than that? Can we learn what's actually different about these different proteins? So these are different proteins. And in fact, prior to picking these, we knew what organelle they were actually in. The human annotators didn't have that information. They're just trying to annotate based on the picture. We know these are actually in 11 different structures. And the question is, could we learn to tell which structure it is just from these images, the task that the human annotators are being asked to do? So what we're going to do is for each of the cells, we're going to segment out what we call puncta. All of these, by the way, are punctate. They are small dots. And we're going to segment them all. And we're going to segment the microtubules, the red lines there. And that's what we're going to use to build our model. And we're going to model the probability that in a cell, there is a punctate organelle at any given position by building a model where we use the microtubules and the distance from the nucleus and the cell membrane to calculate that probability. So this looks complicated, but it's really simple. We're going to say the probability of one of these organelles occurring at a particular set of coordinates given by X is some parametric function, which can be anything, but in this case, we're going to make it really simple. It's going to be a linear function of various factors. So we're going to have coefficients that get multiplied by various factors where those factors are defined at every position in the cell. So to calculate the probability that we have an organelle at a particular position in the cell, we're going to look up these factors that we know the values of the factors at all the positions in the cell. And we're going to then multiply it by some learned parameter coefficient. And that's going to give us the overall probability of observing a puncta organelle there. Okay, so what are these factors? As I was beginning to mention, they're things like how far is any point from the nuclear boundary? How far is any point from the cell boundary? How far is it from how far is it from a microtubule? You really can't see the fine structure of this because I just put, little pictures in here. But this is the distance to the nearest microtubule at any point. This is how dense the microtubules are at any point. And we can do the same thing for the endoplasmic reticulum. So these would be the factors. So if we want to calculate the probability of having a vesicle at this particular point, we take the value of this factor, multiply it by its coefficient, take the value at the same point in this factor, and so on. Okay, does that make sense to everyone? I'll stop for a second there. Of course, we're going to have to learn these coefficients, but we're going to do that with training examples. Okay.
So,
yes,
can you go? Sorry, I'm asking maybe too many questions, but here.
No, there's no such thing as too many questions.
So here you just measure the distance from the nucleus boundary, regardless of the those spots. Is it correct? And then the question is, what is the relation between the spatial distribution of the green spot regarding to the kind of a distribution from for the nucleus, for the ER, or for the microtubules?
Yes. So with training examples where we know the nucleus, the ER, the microtubules, the cell boundary, and the spots, we're going to try to learn to predict where the spots are using these factors. Does that make sense?
Yes. And yeah, I'm curious about the result. And is it if you compare it to a random distribution of the
Yep.
spot or, okay, maybe, thank
Yes,
you.
we will get to that, okay? Now, we just made these factors up. These were things we could calculate from the images, the input images. But maybe you don't need them. Maybe none of them work, or maybe you don't only need one or two of them. So that's a question that we can try to estimate. And here's how we would estimate it. What we do is for all of the combinations of factors. So like we take this and this, or we take this and this, or we take, this and this. For all the combinations, we train a model with just that combination. That means we estimate these coefficients, these parameters. And then we take test data and we estimate the likelihood of the points that are in the test images using the model from the training data. Okay, so we say there's actually a point here in the test data. What was the model predicting was the probability of there being a point there? All right, and that allows us to estimate the likelihood of any test image being from the same model as the training data, okay? And we do that with cross-validation. That is, we take different subsets of the images, take training and train them, and then we use a different subset for testing, and then we change which ones we use for training and testing, and we average the likelihoods over all of them, okay? So here's what ended up happening. We start with a model that just has distance to the nuclear boundary as the only factor. And we get a likelihood of that model. Then we add in the distance to the cell boundary. So now we have two factors, and the likelihood of the model went up. That's better. So we would say it's better to use both of these things than just use the nucleus. Then we add in these two factors, and the likelihood goes up again. That tells us it's helpful to know where the microtubules are in order to reconstruct the puncta. But when we add in instead the ER information, the likelihood actually goes down. That's because it's overfitting. It's trying to make the model depend on the ER, but it doesn't actually. So it does a worse job of predicting, all right? So this is just an illustration of this idea of how we can build conditional generative models. So these are now, we're building a model for the puncta conditional on the nucleus and the cell boundary and the microtubules, but we don't need the ER, all right? So there's a simple version of one of these graphical models. We would say, these are the components, cell, nucleus, and microtubule. We need to know where they are, and if we know where they are, we can then synthesize the punctate organelle, and this bar means it's dependent on microtubule, nucleus, and cell, okay? So that's just a very simple example of that graphical model. Now, how good was this whole exercise? We can assess how good these models are. So we're building a separate model for each of these 11 patterns. So, all right, so for each of these different proteins, we're going to build a model of the distribution of the puncta, okay? And the question is, how good are those models? We can also determine how good the model is by taking some of the images, training a model, training 11 different models, and then for a test image, ask which model gives the highest likelihood that that test image is from that pattern. That's recognizing the pattern. So if the pattern of the test image is actually, the second protein, and the likelihood of the second model is the highest, then the model is predicting this is from the second pattern, okay? Now here's where the deep versus non-deep comes in. The problem is there are only two images in HPA for each of these proteins. for a given cell line. So we do the shallowest possible learning, which is we train with only one image of each protein. So we're given 11 images, one of each protein, and we train a model for each of those using the approach I just told you about. And then we test with each of the remaining images, okay? And as I said, we test by saying for that image, which model gives the highest likelihood, then we say that's what the pattern is. That's what we predict to be the pattern. So here is a confusion matrix of this. So here are the proteins that were called vesicle by the HPA annotators. Notice some of these are not vesicles at all, right? That's coated pits. There's things like retromer in here, and there are even RNP bodies but they look like vesicles, but they're not. They're a subcellular structure. They're not membrane bound. They're not vesicles in any sense, but that's what people think they are because that's the best they can do. So notice, this is the true class of each image, and this is the predicted class from the likelihoods. Notice that the diagonal has mostly high values, right? So the average is here, 83% average accuracy of recognize these 11 different proteins that humans cannot do at all, okay? And you can, back to the question of how does this compare to random? If it's random, you would get 1 over 11 or 9% approximately, right? So the models are much, much, much better than random and much better than people can build. And this is the key, they're learned without having hundreds or thousands or millions of images, okay? So this is another important sort of lesson here or take-home point, that deep learning is great, but deep learning is not great for everything. Okay, so by the way, these are generative models. So we can construct a synthetic cell where all 11 organelles are in the same cell. All right, that's what's shown here. All right, but there are only 4 components in those models, okay? And there are a lot more than 4 things in a cell. And we can't image them all together. So we need to be able to learn models from different subsets of things that we measure and put them together. Now, we showed an example of putting them together by reference to the microtubule. And now we're going to do another version of that. In this case, in collaboration with Seema Lakdawala, who's at Pitt, we're going to try to learn models on subsets, and then with models of different subsets, try to learn how they might connect together. All right, and I'll describe that here. So this is the example case here. I should have paused here before I got to this. Any more questions about the punctate organelle stuff? Okay, so flu virus consists of 8 RNAs that form ribonuclear protein RNPs, and the RNAs are synthesized in the nucleus, then they form RNPs, and then an intact virus needs to have 1 of each of them. Okay, and the question is, how does it get the 8 of them put together properly, right? Does it do it like shown here, that you put 3 together, and then you put, what's the pathway by which this is happening? And this cartoon is just a cartoon. Nobody knows what this pathway is, okay? There are 8 of them, and with the techniques that were available when we did this study, you could only measure 4 colors at a time by fish. And so what this shows is taking images of 4 of these RNPs at a time in the same sample. And the question is, can we use images of 4 at a time to try to reconstruct the order in which they're being assembled? All right. So the first step, similar to the punctate study we just looked at, is we find all the puncta. These RNPs are definitely puncta. They're small, smaller than the resolving power of the fluorescence microscope. And we find all the puncta, and we find, the cell boundary and the nuclear boundary, okay? And we learn point process models like we just did for the punctate organelles, okay? But what we're going to use as factors is not just the cell and the nucleus, but the other colors potentially. So if we're trying to predict this RNP, we're going to learn models that that use as factors the other three and the cell and the nucleus. And then we're going to build all the combinations of those and figure out which ones we actually need, okay? So we're going to build point process models where we're going to get one for each of the 4 RNPs from a given data set, where that model predicts its distribution from some combination of the others. Might not need the others at all, or it might need all 3 of them. Whatever it needs, it's going to build a model for that, okay? And for each of the models, we can estimate the likelihood of the model, how good the model is. That lets us pick what are the good models. And this just shows that what we're looking at here is how strong a predictive model we can build of the distribution of, for example, PB1 from knowing the distribution of M, just pairwise, how well can I predict PB1 from M? And the width of this says pretty well. We can predict very well PA from NP, because this is really wide. Same thing, HA can predict PB1 very well. Some of them can't predict very well at all. So that's just pairwise, but we can do that for 2s, 3s, 4s. All right. So that's the first step. We build all the models because, I should have mentioned, we have many combinations of different RNPs in different image sets. So this set has this 4, then they collected another set that might have these 2 plus 2 others, and they collected not all of the combinations, but lots of combinations of the 8 RNPs taken 4 at a time. So we build a lot of models of those. And then what we're going to do is we're going to do the following. We're going to say, I have models of all of these things taken 4 at a time. Let me try to build a model of all 8 at a time. And the way I'm going to do that is, let's do a simple example if I only had 3 of them. So we're asking, how do we put these 3 things together to make ABC? Do we put A and B together first and then add C? Or do we put A and C together? Or do we put B and C together? What's the order that we do this? Okay. Well, we have the likelihood of various models. This is the likelihood of where B is if I know where A is. This is the likelihood of where C is if I know where AB is. So if I want to, I can evaluate the likelihood of any of these assembly pathways by saying, what's the likelihood of putting together A and B? There are 2 ways I can do it. I can put A depending on B or B depending on A. What are the 2 ways? I'll average them. And then I'll multiply that by the probability or the likelihood that I would have been able to put C properly if I knew AB. And what you can see is that in this toy example, the highest overall likelihood comes if I put A and B together first, and then put C on. Okay, so we're going to use that approach to learn with all the models of the 4s how to put the 8 together. And here's what ends up happening as the most likely tree that would have put them all together. So what it says is, put these 3 together, put these 4 together, and then put them together with this one. Now here's the, we have to think about, well, we were always going to get a tree. There was always going to be the one that gave the highest likelihood, but how much confidence do we have in this? Well, one way we can do that is by adding noise to all of these likelihoods. And if we add noise to the likelihoods, we end up with adding up to 25% noise, which is a lot. We end up with the same tree. So that tells us that's probably the right tree. Okay. So this is an illustration of how we can build another one of these types of graphical models, an assembly model, without actually ever having images of all 8 things together. We can learn them by subsets. All right. So far, we've been talking about organelles, mostly. In this case, we're talking about RMPs. Of course, there are also proteins that are more soluble and, or in complex soluble complexes in the cytoplasm. So we're going to talk about an example of that, again, under this theme of learning dependency. Okay. So this is a study with Christoph Wolfing at the University of Bristol. So here's the problem we're going to tackle. T cells bind to antigen presenting cells in order to, be triggered to, either help B cells or kill cells, whatever it might be. And actin and various regulators are crucial in recognizing that this stimulation is happening. Okay. And so the question is, how are these changing over time and space as when actin is when they are helping to regulate actin? The input data is now just single fluorescence images and a paired differential interference contrast image. Okay, but these are now movies where we have multiple frames of the fluorescence image and the DIC over time, and where time is, in this case, relative to when the APC and the T cell, so this is the T cell, this is the APC, I'm sorry, the other way around, when the T cell connects with the APC, okay? So that's time 0 is when they're touching, or they form what's called an immune synapse. Now, these movies are taken by just mixing together APCs and T cells and letting them fall on a slide. And so different T cells are binding to different APCs at different times. But we're going to define a relative time as when they do this. Okay. The question is, can we build models of the distribution of different proteins during this process? So here's how we're going to do that. We're going to take images that have the APCs and the T cells, and we're going to segment the image into individual cells. So now we have individual cell images, and we're going to align and morph each individual cell to a template, which is a half ellipsoid, which is kind of what the cell looks like, but we're going to make them all look the same shape by morphing them. And that means moving the pixels around until they line up. So now we have individual cells that all have this shape. And then we're going to build a model. In this case, the simplest model is just to find the average and the standard deviation of the intensity of each protein at each pixel in or voxel in this template. So that gives us a model of the distribution of each protein over many cells. We have hundreds of cells over many cells in at a given time point relative to when the synapse formed. All right, and this is showing these models for different proteins at different times after the synapse forms. Okay, so lots and lots of these cell pairs were studied for 10 different proteins in this initial study. That's a higher resolution version of that. And you can see that different proteins have different distributions, right? This is myosin, and it's starting out in the periphery of the cell, but then it comes up and joins the actin distribution. This is an actin binding protein, cofilin, and it moves up into the synapse, 3 minutes after the synapse forms. All right. So this is an example where we can compare different conditions using these models. All right. So what we're going to do is make a very long vector of the concentration of each protein at every pixel at every time point. We have these models at each of the time points. So we have a very, very long vector of all the concentrations at time 0, all the concentrations at time 1, all the concentrations at time 2. We put those all together, and then we can cluster those to see how the proteins are different for different proteins, how they're different from each other. And we can also look at these concentrations to see whether particular proteins are enriching in particular places in the cell at particular times. All right. So here's an example of clustering. So these are the models at different time points. And what you can see is these two proteins are very similar. So this is clustering over that long, long vector. These proteins are very similar. And these are two different conditions where we allow full stimulation of the T cell or when we block half of it. And for this protein, it's not changing its distribution very much. They're very similar. But if you look at some of these proteins like this one, WASP is changing its distribution a small amount from this one. But for example, HS1 here is quite different from its value here. So the ones that are circled are the full stimulus conditions. And what you can notice is that this distance here is how different they are. I think you guys have probably all seen dendrograms before. This is how different they are. So cofilin is quite different for the two conditions. Myosin is very different for the two conditions. And the distribution of wave 2 is very different for the two conditions. So this allows you to see which proteins have a similar pattern over time and which patterns are different over time, which proteins are different over time. Okay. And you can also then look at specific regions to see how the proteins are changing over time. And I'm going to skip over that because it's taking a little longer than I thought it would on this. All right. By the way, this is a movie of those three proteins over time in this template cell. So this is showing the distribution of those during the progression after the time of the synapse formation. All right, so those are examples of constructing generative models using a reference. So in the first case, the reference was microtubules. In the second case, the reference was the other ribonuclear proteins and the cell and nuclear membranes. So we're able to, and then we actually merged the models themselves to construct a larger model. And in the case we just talked about with the T cell, the reference was the synapse of the cell where and we morphed each cell onto the same template so that we can compare them to each other. All right, so that is the sort of the very big picture about the kinds of problems that we are addressing with Cellorganizer. We're going to talk about shape in a bit, but let me stop there. I'm going to open another slide deck in a second and see if there are questions before I do that on anything we talked about so far. We'll hold shape for later. Any questions about how we did the kinds of models we talked about or anything?
So I have just one question about the second model that you explained. So you show that so when you add the here, yes, exactly, yeah, the noise, so we still have the same result. May I ask you what kind of noise or did you add for this model, or how we can?
Right. So it was adding 25% noise to each of the likelihoods. Okay. So when doing this calculation, we would add random noise. You know, we would do the calculation many times. So what would happen is we would sample likelihoods with noise and build them, and then build a tree, and then do it again. Then sample a new set of likelihoods with different noise, and then build a tree. And the tree always was the same. Did that answer your question? Right,
Yeah,
so
just the noise is just a
the
random
noise is
distribution.
Gaussian
Okay.
noise around this likelihood. Okay.
I see. Okay.
All right. So
And
you can
I
see an
didn't
example here.
understand.
I'm sorry. You can see an example here that this likelihood is significantly higher than these. So even with adding some noise, you're going to still get the same tree. Right.
Mhm. Yeah, that my question is, I couldn't understand why this noise makes sense for this particular problem, or for example, just generally, if I want to somehow validate the model, so how can I define, or how can I test validate the model, or create just
right.
Yeah, for example, in the experiment,
Yep.
so we will do some control experiment to compare that the experiment that we are doing is accurate. For example, we add the drug, we have the inhibitors, and something like that. And in the model, if I want to be a parallel pipeline, so we need some validation. And in this case, for example, you added some noise. So is it just, for example, random noise, or it should depends on the model that I want to present?
You're asking a great question, and I'm not sure there's a single answer to it. But in this case, the only input we have is these images and the models we've learned from them. And so the approach we took is to say, well, you know, we're estimating this likelihood. There could be variation in that likelihood. That is, that, you know, we think that we can predict this one with, you know, with this confidence, but maybe we're off by some amount. So, and by the way, the variation in the likelihoods themselves was less than 25%. But what we did is we said, all right, well, let's take a good amount of noise and add it in. And that's just the approach we took to this. And that's the kind of thing you can do often, you know, with modeling exercises and for example, you could say, if you're doing a model in biochemical models, say, well, what if this parameter was, you know, this rate constant was actually more or less by a certain amount, right? And see whether you get the same behavior. Does that answer your question?
Yeah, sort of. I was curious if, for example, I need to add this noise based on for example, the resolution of the images, or the experiment that I'm doing, something like that. But here you added just a Gaussian noise to just change the parameter, or consider within a range instead of just a single value. That makes sense.
Exactly. Now, you're absolutely, you mentioned a good idea, which is that a much more computationally expensive thing to do was we could have added noise all the way back to the images, right? And then reconstructed. But that would take a lot of work. That's why we didn't do it, right, Megan? But it's a good idea, actually. So that's another approach to the same kind of problem. Other questions? So some people are putting questions in the chat, which I would prefer if you could ask it directly, because I can't be watching the chat while I'm presenting. I think I answered the one about poor resolution. Right, so how is the efficiency so good with a single pick for each protein? So I think that was a question. The point there was, it's a single image, but it has you know, something like five or six cells in it. So you have some cells that you can learn. And here's the key about, you know, deep versus shallow learning. Think about how many parameters you're learning in a deep learning model, right? You're learning the values of the kernels, you know, where you're learning, you know, often thousands of kernels with, you know, tens of thousands of values in the kernels. And so that's why you need so much data. In our case, what you're learning is 4 coefficients, because you're given the factors and you're just learning what is the weighting of those factors that I have to learn. So depending on how many factors you're using, that's just however many parameters you have to learn. And that's why it's possible for it to learn with such a small number of cells. So Akash, does that answer your question?
Yeah, that makes sense. Thank you so much.
You're welcome. Okay, let's change gears a little bit and talk. I'm going to go back and forth between the sort of theoretically stuff and some of the more practical stuff. And we're going to talk about a little bit about the paradigms that we use in the construction of Cellorganizer. Okay, so let me give me a second here to open the slides. Okay. So what we're going to talk about now is the, you know, sort of how is Cellorganizer as a software system designed, and, you know, what are the concepts we're trying to use there. Okay. Now the first thing is the original version of Cellorganizer was in MATLAB, and the code, most of the code is still in MATLAB. There are pieces that are in other languages for efficiency, but most of the code is still in MATLAB. But what we also have is versions of the MATLAB code that have been compiled and can be run in different platforms. And the thing we're going to, the most portable, sort of the most useful of those for most people is Docker, which is what we're going to talk about. But there are also versions for other software systems. All right. The main approach used in Cellorganizer is that we have 5 basic functions. This is the API, if you want to think of it that way, where we have this is a typo actually. So we have this function image to SLML that learns a model. So this should say for learning. Then we have mirror function, which is SLML to image, which synthesizes images from a learned model. SLML is historical. It stands for subcellular location markup language, which we actually don't use anymore. We have a different format, but that's what it stands for. So this is the heart of it. This takes images and learns a model. This takes a model and synthesizes images. Then we have 3 helper functions. This prints out some basic information about a model. This is an important one in that it compares 2 models. That's what the report is that you give it 2 models and it writes a report that compares between them. And this is a helper function that will allow you to combine models. So if you learn a model of cell shape and you want, and another model you learn of nuclear shape and you want to combine the 2, you can combine them using SLML and SML. All right, so those are the main functions, and then they take arguments for what to specify their input. So, the models that we learn that can be learned from images in different file formats, which we're going to talk about. Those models are stored in MATLAB files. And of course, it can synthesize images also in multiple formats. Okay, so it can synthesize in, as we'll see, as in SBML, in TIFF images, and more. Can synthesize them in Blender files. Can directly write MCell files and things and various things. Okay, and then as I mentioned, tools for comparing models. So it itself is a library of MATLAB functions, lots of them. It's open source. Any of you are welcome to download and use the MATLAB code. It does not run as a single program. It runs as these 5 functions you would call from MATLAB, or you would call from, as we'll see, from Python in Docker. The MATLAB version runs only in Linux and Mac OS. It doesn't, the Windows version doesn't, the Windows version of MATLAB is missing things that it needs. So we've been talking this morning about training models. The basic idea is what you're going to do is pick some data set that is going to be more than one cell's worth of images. Could be one image, but as long as that has more than one cell, then we're going to be able to build a model. And what tends to happen is that, this is whatever experimental imaging data set that you have. For any of you who are doing imaging and want to use this, you would say, okay, here's the set I want. And that data set should be cells that are all the same, that are approximately the same. So like cells under the same condition, the same time point, the same cell line, because the model that is going to be built is going to be capturing the variation within that data set. So a data set should be something where you expect that all of the items should be built into one single model. Okay. And so like in the example of T cells we were talking about, one data set would be T cells with full stimulation and one protein, and another data set would be T cells with half stimulation and the same protein. So you have now 2 comparable but different data sets that you're going to want to compare between them. So what happens then is that you need to pre-process the data set so that Cellorganizer can read it. Okay, that especially happens if your images are in some proprietary format. At least if that is one that Cellorganizer can't use. And Cellorganizer uses bioformats, so it can read most imaging formats. But if there's one that it can't read, then you have to, convert it into something it can. Most importantly, though, Cellorganizer needs individual cells to be segmented. It doesn't do the segmentation itself. There are lots of packages that will do cell segmentation. And we'll actually use one of those in Friday's advanced topics to show you how to do that. But as part of that pre-processing, the images have to be segmented into single cells. You also need to choose what we call the model class and type, the type of model that you're going to be building. So class would be nucleus or cell or microtubule or a vesicle. Vesicle, for historical reasons, is the category that we lump all the models that are punctate organelles or discrete membrane organelles like mitochondria and lysosomes and so on. So the class, that's like the kind of structure you're modeling. And then the type is how the model is being built. And we're going to talk about a couple of different ways of building models of the same thing. So you can have a model of cell shape that is learned with one method, and a method of model of cell shape that's learned with a different method. So the class would be cell shape, and the type would be whichever method you're using. And then, very importantly, you have to select some options that might be needed for the particular type of model that you're building. And we're going to show examples of that. And you may need to select some options to tell the system things that about the images themselves. For example, to tell the pixel resolution or voxel resolution of the images. If it can't read it for itself, you have to tell it so that it knows the size of things in the image in order to build a model. And so that's done by setting various options in what we're going to see as an options block to give it information about the image that it can't figure out for itself. All right. So here are examples of class and type. So Cellorganizer can build either 2D or 3D images models. For 2D, there's much less work on 2D images because 3D are much more interesting, and you can build much more accurate models from 3D images. So these are the classes right here as the ones I mentioned, nuclear and cell membrane. There's also a category called framework, which is a paired model of the cell and the nucleus, dependent on each other. Here's an example where you can build a framework model using a diffeomorphic method, or you can build it using a PCA method, and we're going to talk more. These are shape methods. We'll talk more about them later. But this is just an example of how you would say, all right, I want to build a model of cell and nuclear shape. Therefore, the class model I want to build is framework, and I want to use PCA for that model. And so I set the option for class to framework, and I set the option for type to PCA. And what I'm going to get is a model that captures both cell membrane and nuclear membrane. Okay. For 3D, there are a lot more. As I mentioned, we have cell nuclear framework. We have network model for microtubules. We have model that is just protein distribution like we used for the T cells, where it doesn't have any discrete organelles. It's just a distribution throughout the cell. This is just a special model that's used for this half ellipsoid. It's there because we need it, but it's no, they wouldn't use it for anything other than the T cell models. This is constructive solid geometry objects. So you can actually construct arbitrary ellipsoid models from this. There is also then these vesicle categories. And we're going to talk a lot about this later. So this is, and then, depending on what the options are set here, then you get the corresponding kind of model. All right, so what do we need to provide to image to SLML? We need to tell it, is it a 2D image or a 3D image? We need to give it the path to the images, and we need to give it the options. And then it's going to produce a model file. It's also going to produce auxiliary files that have the parameters for all the images, and we're going to take a look at that when we actually do the tutorial later. So I mentioned already that the images are, depending on what kind of model you're building, you obviously have to have the structure that you're trying to build a model of labeled with something if you're going to try to build that model. Now, as I mentioned, dimensionality is the first argument. And then the way it's set up is that it takes always 3 sets of images. Images that define where the nuclei are, which might be a DNA stain. Images that define where the cell boundary is, which might be a membrane probe, or could even just be an autofluorescence image, or might be, some soluble cytoplasmic probe. And then what we call the protein image, which doesn't have to be a protein, it can be a fluorescent probe as well. But it's the thing that is going to be built conditional on the cell and the nucleus. Okay, so the input is then going to be 3 sets of files, and you notice that we have a wildcard in here, right? So this is saying for the DNA images, take anything that's cell, anything DNA. That's going to be your DNA images. This is going to be your membrane images. This is going to be your GFP, your protein image. And then we're passing in also an option structure. So as I said, each image has to be, each cell has to be segmented. So you can do that 2 ways. One where you have separate files for each cell, or you can have an image with multiple cells and either create a mask that tells you where each cell is, or the best way to do it is in an OME-TIFF file to specify regions of interest that define each of the boundaries of the cell. I'm going to pause there to see if there are questions about that, because people may, or anything we talked about. I should have paused once earlier. Questions about any of that?
May I ask a question?
Mm-hmm.
So this is about earlier when you were talking about the model where you predicted the cell organelles. So if there's a case in tissue like that, locations of the proteins are not well predicted by what is in the cell itself, but by the cell next to it. So there's a cell-to-cell contact that better predicts where those proteins are. How would that be done here?
Yep. So that would be a case where you would need to use the code that is underlying Cellorganizer directly. What I would think that you would want to do there is a point process model, like we were talking about earlier, but where the factors would be distance to the other cell. Does that make sense? So
yes, but would you?
So
how would
and now
you
what
need
would
to
happen
segment these cells as together, like clusters of cells, or do you still segment it individually?
Right. So you could. There are a couple of ways to do it. One would the probability map is in the cell that you're trying to synthesize, right? So, but the factors can be distances to things outside the cells. So you would have your individual cells that are the ones that you're trying to study the protein in. And then you would just provide the factor matrix of the distance to the other cell in that. Okay. So there would be a way to do that by passing in that factor directly in the options.
Okay, interesting.
Okay.
Cool.
But you would have to learn the factor outside. You would have to measure it outside.
Yes.
Okay. If you're interested in that's an example of something we'd be very happy to help with. Other questions?
I have one question.
Sure.
Over here, all the inputs are from experiments. Is it possible to take inputs as screenshots of a simulation?
Okay, interesting. So you mean to learn a model of something like the distribution of a protein from simulations of the diffusion of the protein? Is that the kind of thing you're thinking of?
Right, right.
Yeah, absolutely. The input can, if the input is an image, it's going to work. Right. So you would just have to make sure that you get those simulated distributions into an appropriate image format. And if it's if it's a model over time, then you would need to associate the time with each of them and so on. Okay, yeah, absolutely.
Makes sense. Thank you.
And you could also use it to distinguish between the pattern of two different simulations, right? So you simulate with one parameter value in your simulation, and then you do a different one and you compare the two to see what's different. Make sense?
Right. Yeah.
All right. Other questions? Okay. So the images need to have single cells, or they can be multiple cells, but you have regions of interest. The images also have to have the different fluorescence channels, right? The channel that is showing where the nuclei are, where the cell membrane is, where the protein is. The assumption is that those are in separate files, but you can also have them in the same file, which is much more common in actual imaging. But when you specify the images, you put a colon in the channel that you mean to use for that particular purpose. Okay, so in this example here, if we had all of these in the same file, let's say that the file spec would be this, we would say :1, that's the DNA channel, and then maybe :3, that's the membrane channel, and :2 over here for the protein channel. But the file names would all be the same. All right, and we would just specify which region, which channel we want from each image. OME-TIFF, if you haven't seen it before, is a very well supported format for storing images, multi-channel, multi-cell images, and there are a lot of tools for OME-TIFF, and we're going to use that on in the advanced topic session on Friday. Okay, so this is the most complicated part of Cellorganizer probably, which is setting the options the way that you want in order to do what you want. In the MATLAB version, it is a structure. In the Python bindings of this, it is a dictionary, Python dictionary. And it has many options within it that, as I mentioned, there are options that tell us something about the input images in case that's not already in the image. There are options that will, for example, allow you to downsample the images before you model if you are worried about the amount of time, the compute time it would take. If your images are very large, then you might want to downsample. Or if you just want to examine the effect of different resolutions on the model that you build, so you can specify downsampling. And of course, as we said before, you specify the specific type of model that you want to build. And if it's not an OME-TIFF that already knows the resolution, then the resolution is mandatory in that case. Otherwise, it doesn't know what to do. The output is a MATLAB binary file. It has everything it needs to reconstruct, to synthesize a new image, and it has a lot of other stuff that it's saving inside of it. As I mentioned, there is a tool for getting information about a model that's called SML-info, and it just gives you back the content, parses the contents of the model for you. It will also do various things, like if there are any reasonable plots that might be, that you might generate. SLM report is very useful for comparing two different cell types, two different conditions, basically any two models that you want to compare. However, the models need to be of the same type, right? Same class and type. And as I mentioned, we have the tool for joining models, okay? Once we have a model, we can generate images from it. And as I mentioned, those can be various formats, and including SBML spatial, which is widely used, okay? So, here are some example, that's an example of synthetic images. So, as I mentioned, MATLAB, the code is MATLAB code mostly. what we have done is compiled those 5 modules. So it's possible in MATLAB to compile a particular function. And now what it produces is an executable image, executable file that runs that particular function. And that MATLAB requires a license. Running that executable file does not require a license. It does require the MATLAB support libraries. Those are, you can copy those without having a license. So what we have done is we started by compiling those 5 binaries. And then we made a Docker container that contains a Linux OS and the Cellorganizer binaries and the MATLAB libraries that the binaries need, and a set of Python functions that can call the binary, the executable files. You can run the executable files yourself if you want to write your own interface. You can run a shell script that would run them, but the easiest way to run them is via Python. All right. I think we can skip over that. It basically, it's making, the only issue is there are a very few things that you can't do in the executable version. But as you'll see, most of the things you can do. And so so this is just summarizing. I forgot I put this in here, right? Cellorganizer has Cellorganizer and the MATLAB libraries and Anaconda and Jupyter Notebook, so that you can create Jupyter Notebooks that can run Cellorganizer. All right. I think I've talked about all of this stuff. The difference here is that the options are now a Python dictionary, not a MATLAB structure. We'll see that later. And as I said, you can run it through Jupyter or through separate Python or shell scripts. When you download it, when you pull the Docker container and start up the container and run Jupyter Notebook, then the way we have it set up, it will connect to a folder on your local computer so that you can access files on your computer through that folder. And there's also something that will pull all the files you need for the workshop. And so last thing I want to mention about that is, as we'll see later when we're running it, the way that the Jupyter version works is that you start up Jupyter and you're interfacing to Jupyter to run things. But once you start running something, the output that Python is generating. So I mentioned that we have Python wrappers for each of the 5 functions. The output that any of your Python code is generating, like here's a piece of Python that is, says print this. Well, that of course shows up on your notebook. But the output that is coming from the MATLAB Cellorganizer code goes to whatever terminal window you started Docker from. Okay. And so in many cases, you would execute a command and it would just say, yep, I did it. You would get, nothing but, success or something is your message on the Python window. But there may be a whole bunch of output of all of the things that illustrating all the things it did while it was running. And if there are problems, typically then you find whatever error happened in the terminal log. And then all the files it's creating, of course, get created in whatever the default folder was when you ran the notebook, or whatever the Python command set as the location. All righty. Questions about any of those kind of basic logistics of running it, or sort of the paradigms of the different modules and the inputs and so on. If not, we're just about on time and I'm overdue for lunch. So we'll take a break and resume at, I think it is in, well, at 15 after. Correct?
Yes. Yes, correct. Thank you very much.
Okay.
So see you in 15 minutes.
Bye, everyone.
And please use the new Zoom link. So we are going to use another Zoom. So I should close this one. Okay?
All right. Bye, everyone.
Thank you. Bye-bye.